The control mechanisms on the cell cycle. Let's learn some more details about the cell cycle control. Now, first of all, there are two groups of proteins you have to learn before you start trying to understand cell cycle. Those two groups of proteins are one, if I can write here, one, cyclins, cyclin proteins or cyclins. Two, cyclin-dependent kinases, CDKs, CDK, which means cyclin-dependent kinases. So, first you have to learn these. You have to learn the names of these two proteins. Now, we have two groups of proteins. One, cyclins. Two, cyclin-dependent kinases. Shortly, we call them CDKs. There are a lot of cyclins and there are a lot of CDKs, cyclin-dependent kinases. As you can understand from their names, CDKs are enzymes, kinase enzymes. So you should remember the kinases from our previous lectures again. Kinases are the enzymes that phosphorylate the other proteins or other molecules. And they receive the phosphate group from ATP. So those are ATP-dependent enzymes. They remove one phosphate group from an ATP and bind it to their target molecules. So kinase enzymes bind the phosphate group to their target proteins or other molecules. So CDKs are kinase enzymes which requires other proteins to be activated. Those proteins which CDKs need are called cyclins, okay? So we have a group of enzymes. Those are kinase enzymes. They phosphorylate their target proteins, so they activate them. But these proteins need another kind of proteins. Those are called cyclins. Because of that, we call these kinase enzymes cyclin-dependent kinases. And the proteins which control them, cyclins, okay? So these are two important groups of proteins for cell cycle control. Let's go back to our screen share. All right. Cyclins and cyclin-dependent kinases. Two types of regulatory proteins are involved in cell cycle controls. Those are cyclins and cyclin-dependent kinases, CDKs. The activity of a CDK rises and falls with the changes in concentration of its cyclin partner. Now, this cyclin concentration, the concentration of cyclins in a cell is not constant. It's not always at the same concentration. What does it? The concentration is concentration of these proteins in the cell. So in one cell, how many molecules of that specific proteins exist? For example, how many cyclin 1 is there in a cell? Now, the concentrations of proteins, especially these proteins, are not constant. Or let me say that the concentration of CDK in a cell is constant, but concentration of cyclin is not. So to increase the concentration of cyclin or any other protein, to increase the concentration of a protein in a cell, what does a cell have to do? It has to synthesize that protein. So we are going to protein synthesis. So the cell synthesizes a protein. This protein can be a cyclin or any other protein. Do you think once a protein is synthesized, it's always there? It always stays in the cell? If you think so, it's not. Once we synthesize a protein, that protein stays for a limit. That protein is usually those proteins are usually exist for a limited time. So after a time, of this time period, after a time period, this time period can be a few minutes, a few hours, or days, or even some proteins can stay for weeks and weeks in the cell. But some of the proteins have shorter lifetimes. So every protein molecule has a half-life. After we synthesize a protein, after a cell synthesizes a protein, it also degrades that protein too. Degrade means degradation to hydrolyze the proteins, to break the peptide bonds between the amino acids of protein and separate the amino acids of protein. That degradation. Now, a cell can produce a protein, a cell can synthesize a protein if it needs, but that protein, some of the proteins are degraded by the time, and this time, by this way, the cell adjusts the protein concentration in the cytoplasm. So by synthesis of protein, that protein's concentration increases, but by the time, while that protein is degraded by other proteins in the cell, the concentration of that protein decreases. So some of the proteins have constant concentrations in the cell, but some of them do not. So in this example, in the cytoplasm, there are always same concentration of CDKs, but concentration of cyclines change. There's another term here, MPF, maturation promoting factor. MPF means cyclin CDK complex, because CDKs require cyclins to be activated. So a CDK is active only when its cyclin partner is bound to it. This complex is called MPF, maturation promoting factor. This cyclin CDK complex triggers a cell's passage the G2 checkpoint into M phase. So there is a cyclin CDK complex, which is called MPF. That is the molecule which helps the cell to pass the G2 checkpoint. Okay, now we have learned the first cyclin CDK complex, which is called MPF, and now we know better how the cell passes the G2 checkpoint. All right. This is the symbol of MPF. Okay, so when cyclin is dependent to its cyclin-dependent kinase, when these two proteins are bound to each other, the cyclin can be activated. The cyclin is, the CDK is active. But if the cyclin is not bound to CDK, CDK cannot work. It cannot catalyze its reactions. That means it cannot activate the other proteins. So if we look at the protein concentrations, the cyclin concentration and the CDK activity, let's look at the CDK activity first, or MPF activity. MPF activity starts by the start of mitosis, and at the end of mitosis, it loses the activity. And in the next cell cycle, in the next cycle of the daughter cell, again, we can see the CDK activity only by start of the mitosis, and activity of this kind of CDK ends at the end of mitosis. And in the next cell, so the cell has divided, there are two cells. The next one again shows the activity of this CDK, this kind of CDK only during mitosis. Then there must be something triggering the CDK activity. That is cycline concentration. So let's look at G1. At G1, there is no cycline in the cell. At S phase, there is no cycline too, but at S phase, at synthesis, at the S phase, during the S phase, the cell, I said, synthesizes some proteins as well as the DNA. So one of those proteins is this cycline. The cell starts synthesizing this protein during S phase, and by the end of S phase, during G phase, during G2 phase, while the cell is synthesizing this protein, concentration of that protein increases in the cytoplasm. And after a concentration, concentration point, these cyclins are able to find the CDKs, their partner CDKs, and they bind to them. But for this, they have to reach to a concentration in the cell. When the cyclin binds to a CDK, that CDK molecule is activated. When another cyclin binds to CDK, the second CDK is activated too. So by the increase of, while the concentration of cyclin is increasing, more CDKs are activated. And this activation is at the top, by the middle of mitosis. And this increase causes the cell to pass the G2 phase. So this is one of the mechanisms that check the G2 checkpoints. After the cell completes its division, the cyclins in the cell is degraded, and during the G1 phase, no more cyclins are synthesized. So CDKs in the cell are inactive. In the next cell, in the next cell cycle, by the middle of S phase, the cyclins start to synthesize, the cyclin concentration increase, and after they reach a point of higher concentration, they start activating the CDKs, and that takes the cell to M phase, okay? But this is only one cyclin CDK complex. There are other cyclin CDK complexes too. In this picture, you can see better as well. So this is G1. This is G1 stage. There are CDKs, cyclin-dependent kinases, but there is no cyclin. So CDKs are inactive. When the cell passes the G1 checkpoint and it goes into S phase, during S phase, this cell starts to synthesize cyclin. So this arrow shows the cyclin synthesis. While the cell is synthesizing the cyclin, that causes cyclin concentration to increase, more cyclin proteins to be exist in the cell, and after a point, these cyclin proteins are able to find the CDKs in the cytoplasm, and when they find the CDKs and when they bind to them, CDKs are activated. Activation of CDKs causes the cell to pass the G2 checkpoint and move into mitosis. And by the end of mitosis, cyclins are degraded. By the end of mitosis, cyclins are degraded, and they are removed from the CDKs. Now CDKs are inactive. When? By the time the cell completes mitosis. When the cell completes mitosis, cyclines are degraded, CDKs are empty, they are alone, and they are inactive. So did you understand? Stop and go signals. Internal and external signals at checkpoints. So checkpoint means the time points that the cells receive the stop and go signals. Many signals registered at checkpoints come from cellular surveillance mechanisms within the cell. So most of them are intrinsic. Checkpoints also register signals from outside of the cell. 3 important checkpoints are those in G1, G2, and M phases. For many cells, the G1 checkpoint seems to be the most important checkpoint because that's the point in which the cell decides to divide or not. If a cell receives a go-ahead signal at the G1 checkpoint, it will usually complete the S and G2 and M phases. If the cell does not receive a go-ahead signal, remember the growth factors, it will exit the cycle and switch into a non-dividing state which is called G0 phase. G0 I already explained. All right. So in these pictures you can see which one should I show? All right. So this is the this is the whole picture of cell cycle. In a cell, which is here, a cell is at this point at G1 stage. It has two choices. The cell can choose the first way, not to divide. Then the cell goes into G0 state. Without a go-ahead signal, the cell enters to G0. Without go-ahead signal, the cell cannot go to, there's something wrong here. Okay, let's go. But if the cell receives a go-ahead signal, then it passes G0, it exits from G0 and it passes G1 and it starts to S phase. And during S phase, we know what happens. So you can think these are go ahead or stop signals. So actually there is a go ahead signal, there are go ahead signals which are growth factors. And without presence of growth factors, the cell cannot exist from G0. An example of an internal signal is that cell will not begin anaphase until all chromosomes are properly attached to spindle at the metaphase plate. You remember this, I already told. This mechanism ensures that daughter cells have the correct number of chromosomes and they are attached to the spindles. Growth factors are the external factors. External factors that influence cell division to cause cells to divide. So these are growth factors. Growth factors are released by certain cells and stimulate other cells to divide. So there are two different kinds of cells. One of them produces growth factor, and that growth factor causes the other kind of cell to divide. Example here is the one I told you a few minutes ago, platelet-derived growth factor, PDGF. PDGF is made by blood cell fragments called platelets And in density-dependent inhibition, crowded cells will stop dividing. Okay, this is a picture showing the cell culture that's possible to produce mammalian cells in the laboratory in vitro conditions. A sample of human connective tissue is cut up into small pieces. For doing this, we take a sample of human cells, like biopsy. We put it into a petri dish, and we add the enzymes that digest extracellular matrix to separate from the cells from each other and suspend. And then we put these cells into flasks. These are flasks. These special bottles are called flask. Then after a time, they grow. But if we add growth factors, if we do not add any growth factors, they cannot increase, they cannot divide. But if we add growth factors, then we have a lot of new cells even out of the body. So in this SEM picture, electron micrograph, you can see cultured fibroblast cells. These are human cells. Okay, in here there is an important thing, anchorage dependence. Did I tell you about this? Have I ever told you about anchorage dependence? Do you remember this? Anchorage dependence daha önce hiç bahsettim mi? All right, anchorage dependence, most cells also exhibit anchorage dependence. That's another mechanism that controls the cell division. To divide, they must be attached to a substratum, which means a solid surface. So without binding to a solid surface, the animal cells most of the animal cells cannot divide. Density-dependent inhibition is another one. Density-dependent inhibition and anchorage dependence check the growth of cells at optimal density. So, for an animal cell to divide, first, it has to bind to a solid surface. Two, there mustn't be other cells touching to the cell. These are density-dependent inhibition and anchorage dependence. Cancer cells exhibit neither type of regulation of their division. So normal animal cells do not divide if they are not on a solid surface or if they are surrounded by the similar cells, but this doesn't work for the cancer cells. Cancer cells don't care if they're on the solid surface or if they're surrounded by the similar cells. So density-dependent inhibition or contact inhibition or anchorage dependence laws don't work for cancer cells. That's why they are cancer cells. All right, anchorage dependence, the cells require a surface for division. So once we put the cell into a flask like this, and when we add the medium, growth medium, the cells find their places and they stick to the bottom of the flask first. Then they start to divide. But until what time? Until when? Until the flask is filled up, bottom of the flask is filled up with the cells. Once the flask is full, all cells are in contact with the other cells, they stop dividing. This is contact inhibition. So at this step, the cells stop dividing. But if we take off some of these cells, if we make an empty area, the cells around that empty area start dividing again, and that fill up this empty space. This is called density dependent inhibition or contact inhibition. This is not seen in the cancer cells. So by this way, the cells make only one layer of the cells at the bottom of flask, cell culture flask. But if these are cancer cells, they keep dividing even they fill up the bottom of flask because they don't care contact inhibition or anchorage dependence. All right, these are normal mammalian cells in the culture medium, but these are cancer cells. So even morphologically, you can distinguish the cancer cells from normal cells.